So your notes right now are looking at the mathematical analysis of data. We're getting ready to design experiments. And so we really want to think about the scientific practices. And we want to start by going back to think about what is the point of an experiment. Experiments are really fun, but why do scientists actually perform experiments? And so if we think about the value of an experiment, really what's happening is that we are gaining knowledge. We are trying to use a series of steps. We're trying to use a method, so the scientific method, to gain knowledge of certain conditions. So when you think about what an experiment is not making a paper mache volcano where you add vinegar to baking soda and it explodes. That is not an experiment. That is a demonstration. An experiment looks at different conditions. And so we're going to need to be able to vary these conditions. And as a result of these varied conditions, we're going to gain knowledge. And the overall goal is that we want to be able to apply this knowledge to a real problem. So even though we're going to be looking at roly-polies and we're going to be choosing conditions that may not have a huge impact overall, if you are a scientist proposing an experiment, looking for a grant, your grant has to have practical application. You have to be able to apply this knowledge to some real problem that you're trying to solve. We do want to take a second and talk just a little bit about the different types of experiments. The ones that you have most likely done up until now are what we call cookbook labs. And this means that they give you specific directions, and the outcome is known. Versus what we're going to be doing this year, which are called inquiry labs. Inquiry labs are going to be where you're creating the steps. And the outcome is unknown. Just slow down when I write. Unknown outcome. And a lot of this time, this means you may or may not get perfect results. Sometimes you have to repeat an experiment over and over and over again. When we look at what the College Board or AP Biology curriculum has to say, the focus of the labs has shifted from students achieving predetermined results to students making independent observations, posing their own questions, and determining how to investigate these questions. And that's where inquiry comes into play. But a big part is going to be how do you collect the data? And that's what we're really looking at in starting the year by letting you just design an experiment, a beginning experiment. So really get you start thinking about how do I investigate a question? How do I collect data? Now let's keep in mind one of the big things you want to do is collect lots of data. So we've reminded ourselves why we do experiments. We're trying to gain some sort of knowledge that we can apply to a real world problem. But we actually want some sort of valuable knowledge. And what that comes from is collecting data. Being able to have actual information at the end of an experiment. So next we need to look at what types of data can be collected. And so when we look at data, we really want to understand where does this data come from? What is the origin of the data? Might be the first thing. What data do we mean? So let's go back a little bit to the scientific method. And remember that you are going to be varying the conditions. And what you vary is called the independent variable. Independent variable. going to be how you vary the conditions. So for example, with the roly-polies, you might vary the temperature of the environment. Now, that will be preferably a number. Preferably you won't say hot or cold. Preferably it will be a number like in degrees Celsius. And those are data points. But what we're more interested in is what is the result of changing this condition. So the dependent variable this is going to be what we measure as a result of the independent variable, IV, independent variable. So you're going to change something like the temperature, and then you're going to measure something as a result. So if you go back to the top of the page and you look at the little pictures of the pill bugs on the left side or on the right side, we can actually count how many pill bugs, isopods, roly-polies are on either side. So the number, this might look like the number of isopods on each side. And so when we set up an experiment, we will be varying something, and preferably we'll be varying it in specific intervals, and as a result of these changes, this is the data that we're really interested in, the dependent variable data. So I can change whether I measure it at 0, 30 seconds, 1 minute, or a minute and 30 seconds, and I can change whether it's wet or dry. But what I'm really interested in is the number that are in the wet and the number that are in the dry, or the number that are in the hot and the number that are in the cold. Now, just a little aside, we do want to remind ourselves about a control group. Now, preferably, you would collect lots of data. So let's say we're working with temperature, we might do 5 degrees Celsius, 10 degrees Celsius, 15 degrees Celsius, 20 degrees Celsius. You would want to have a lot of different independent variables. But one of those independent variables should be a control group. Now keep in mind, we only really want to change one condition at a time, but we want multiple intervals of that condition. So your control group should really be your normal conditions. And this is used for comparing. If all I do is put the bugs in wet, I don't really know how that compares to their normal condition. Now, a big question is their normal condition dry? So this is where we'd actually need to do some research to know what is the normal condition for an isopod. So when we go and we look at a lot of data that we have collected, we want to keep in mind that this data can be separated into qualitative or quantitative data. Quantity are numbers. So if you can give it a number, then it's considered quantitative data. If you're more describing the quality of it, so a lot of times color is thought of as qualitative. Texture is thought of as qualitative. You might be able to create a gradient, but you can't necessarily give it a number. What we're going to see more of this is in terms of behavior. So this is where these other notes come into play. So we might have seen that they are climbing the walls. And that is going to be a qualitative piece of data. And that qualitative data is valuable as well. But you can't really analyze qualitative data. So we focus on quantitative data because it can be analyzed. Using mathematical tools. Math and statistic tools. And so that's going to be part of our job this year, is being able to analyze our data. Quantitative data actually comes in two forms. The first is what is known as measurement data. And the second is known as counting. In counting, you have discrete numbers, like whole numbers. So 1 isopod, 2 isopods, 3 isopods. That's it. We have a discrete number, and that's more counting. Measurement is where it's going to be more continuous. And this could be, for example, 1.1, 1.01, 1.001. This is where significant digits will come into play. Measurement data is actually much more valuable because this is measurement equals continuous. Measurement data are continuous, and this is much more valuable because we can look at the distribution of data. And so we will this year be using a lot more measurement, but we're starting this year by counting. We're going to count the number of isopods. So we've collected data, but the College Board says, what does this really mean? Does this represent a useful or meaningful result? How can I test this? And we do want to remind ourselves that we need to be able to repeat this. So when you're designing your lab, you need to keep track of what it is exactly that you did so that you could repeat your experiment. So I do want you to take just a second to, we're going to talk just a little bit about some statistical analysis, but we're going to get much more into this later in the year. It's really important that we start to use some specific statistical tools because statistical analysis makes it more significant. It gives more meaning to it. And one of the reasons that it does this is that we can examine the data for causation. I just spelled causation. Yep, the U goes first. Causation versus correlation. Now, we can do our best analysis on measurement data, not counting data. So that's something to keep in mind. Causation is where we can say there is a deliberate relationship. So causation means that the independent variable caused any change, delta, this triangle means change, in the dependent variable. Correlation means that there is some sort of relationship, but we're not quite sure if it's a direct causation relationship. And so we're going to be using tools to actually be able to support causation. The last thing that we want to do, and if you look at the additional document you were given here, the AP formula, it'll show you some of the stats that we'll be doing this year. But if you go back, the last thing that we really want to do is look at hypothesis. Now, a reminder of hypotheses that you have written is if the independent variable, so if pill bugs are placed in water, if the IV ... then the DV ... Meaning if I change this, then this will happen. And this is actually going to be because. But we're going to look a little bit more about something called a null hypothesis